And to discuss stem cells and sex, we need to define at least two things: stem cells and sex. Yes. I'm sure that you intuitively know what a stem cell is. But I want to add that our focus is human today, and we will talk about human development. And I think animal studies are questionable for human studies on human development. And when we talk about perception of human gender, I think animal experiments will be completely useless, according to my view. So, but the definition will be the same. A stem cell is an undifferentiated cell which has not yet decided what to become in life. It's not committed to any specific function or path. And it can self-renew, which means that it can divide, multiply itself to more of itself, and keeping this undifferentiated state. And it has developmental potency to specialize into several different cell types in the body. So these are some of the questions I will address, and our focus is still human. Like I said, other species have other solutions to what we're going to talk about. Stem cells are very important. They're important for almost every step of the way of development of our bodies, and from the first stage of the embryo up to the adult. We will walk through these stages of stem cells types, and pay attention to the impact of sex and sex differentiation. And of course, you heard a story about bees and flowers before, but maybe not my version, and maybe not focusing on stem cells. So I will endure with me. I will try to explain. This is, in a way, a complement or an addition to what you heard yesterday from Londa Schiebenger, who spoke about gender innovations and cell. There is a website that perfectly well matches what I'm saying, and you can add things from my lecture to that site, and maybe some of the references we can maybe exchange them later on. And I realize that you will not get my lecture, but if you contact me, I will send it to you in a PDF form, and you get the references. So if we go through this slide, the start point is the egg, which is newly fertilized. It's called a zygote. And this is a special cell. That's the mother of all cells in the body. It's the stem cell. This cell can become everything. It can become every cell in the body, and it can also become cells that are extra embryonic, that is, supportive cells like placenta. This stage is kept all the way up to the 8-cell stage, and I will explain in a moment why I picked the 8-cell stage particularly. But those cells are called totipotent, and it's easy to understand why then. After that, an important stage is the blastocyst, and that's of course because this is where you harvest embryonic stem cells. And embryonic stem cells are also very potent, developmentally potent. They can become everything, but they cannot become placenta according to the dogma. So that's reassuring when you work with embryonic stem cells, that you cannot make a whole human being of them. But that maybe is not completely true. After this stage, there is an implantation and a fetal stage. And during this stage, you will also find stem cells of different sorts. And in the beginning, they're also pluripotent, and eventually they will become multipotent, which is a little bit less potent. They can become a little bit less. There is a reduction of potency here. And I might add that there is this size from the egg or the zygote up top to the blastocyst. They are the same size. That's because we have a shell. And like chicken or hen, we hatch. When this shell is too small, we will burst out like an amoeba and climb out of this. And this sticky little creature will attach to the womb or the epithelia of the uterus and implant like a cancer and invade the woman. And this is really a cancer, similar to a cancer trait. I will also say that the embryo up to this stage of implantation lacks oxygen, so it uses sugar for energy, and that's called glycolysis, and that is used by cancer cells for energy. They don't use oxygen in the same way, and the embryo up to this stage is very similar to a cancer in this respect. To sum up this slide, stem cells are indeed very important and for development. In fact, they make it happen. And furthermore, if things go wrong, you get a disease. And that could be, like you see on the bottom of this slide, cancer also got stem cells, and they also have potency. So if there is a link of stem cells to sex, then there is a link for sex to cancer, and there are different cancer perspectives, and we should definitely talk about these things. And like you discussed with me over the email, the metabolism of these stages are similar to cancer, so this is definitely a hot topic to look at metabolism in stem cells. Furthermore, okay, there is a link to health and disease, and if there is a link to health, there is a link to financial industry or funding or money. And if there is a link to money, there is a link to politics. And if there is a link to politics, there is a link to ethics. And if there is a link to ethics, there is a link to religion. And we end up in a very emotional situation. So stem cells are definitely of importance to many aspects of society. Now, anyway, I would like to remember one thing. Remember, please, that the first stem cell was a newly fertilized egg, the so-called zygote. Please keep that in mind. Regarding sex, the word sex can mean a lot of things. And the purpose of, as I understand it, is to drive occurrence of new combinations of genes in our children. And this way will enhance the chance of species surviving in new conditions. That is, the children will have, at least one child will survive somewhere in the new condition. And that's a species advantage. It's not necessarily an advantage for the individual or the family, actually. You do get new genetic combinations, and not all of them are fortunate, so actually you can as well get a sick child while you're healthy. But the bottom line is that we have two sets of genes, one from mother and one from father. And let's for a moment define sex as a presence of X and Y chromosomes. This is genetic definition. I hope I'm in time now. So now, of course, you remember what I'd ask you to remember, right? The first cell, stem cell, is a zygote. And in order to form this, we need sperm and we need eggs, and they are haploid, that is, they will together make the full genome. And this is a start, so already here we find the first differences between male and females. Females start, each progenitor cell or stem cell make one egg. And all eggs are present when you are born. You have a couple of 100,000 eggs when you're born, and you won't get more. And every month from puberty, you will mature one, or sometimes twice. The older you get, sometimes twice. And you will run out of them, and they will all be used up at menopause. So that is the fate of the woman. Man has a different situation. One stem cell will make 4 sperms. Spermatogenesis starts at puberty, and large numbers are produced throughout life. So that's a big difference. And men are much more living in the moment, I wrote, in this way. There's quantity rather than quality. And we're active all up to ages until we die. And this leaves us with differences in risks, actually. Environmental risk dangers are different from dividing cells and non-dividing cells, like irradiation and chemicals in our environment. So we have different sensitivities for these stages. Let's go back to the zygote. We're not quite yet ready. After the sperm has penetrated the egg, initially the genome of the mother and the father are not mixed. They are sitting next to each other. They are moving towards each other, and eventually they will fuse. This is a story of time. It takes up to actually 3 cell divisions or 3 days before the male genome starts to talk. It's completely silent for the first 3 days, for the first 3 cell divisions. It's a female genome that drives the whole process. Total control. It's called transition, when the embryo is taking over its own life and caring for its new unique DNA for the first time. That's maybe why the egg is so large, because it carries material, nutrition, information, messages, which the male cell doesn't have. So the female genome takes full control and take care of food and growth in the beginning. No other comparisons. I will not make that. This means, importantly, that conception takes 3 days. And this is more important than you may think of politically. Tell that to the Pope. And ask him to reconsider about the day after pill. I have spoken to Jesuit paters about this in Sweden, and he promised to bring the message to Vatican State. But there is this fact that conception is not overnight or a moment. It's 3 days. It's just a thought. Anyway, after 3 days, we have a new individual which has mother and father DNA and a new fresh start. Let's talk a little bit about control. The use of genes are controlled. And think about the genome as a piano. You can play on the same piano rock and classical. It's just a matter of how you use your genes. The previous control of the parents is erased at the time of when the gametes are made. It's like pressing reset on a computer. So the baby starts fresh and starts its own control. And this is settled at the stages of gametogenesis. Now come the next differences, and I would envy your picture of the X and the Y chromosome. I couldn't find such a nice one. But the X chromosome is a lot huger. It has, no one knows exactly how many genes, but about 900 genes are transcribed into proteins. And the Y chromosome, 50 to 60. That's a huge difference. It's about 3 times more long, the X chromosome, than the Y chromosome. And that, of course, makes a difference, a whole chromosome difference. Which means we have 2 sets of DNA, and we need them to choose which one to use, actually. And nature has provided a mechanism for that. It's called imprinting. And you use either the maternal or the paternal gene of a special gene combination. And this is actually a new knowledge, only 30 years that we have known that this is the case. But also you have 2 X, and immediately one of the X chromosomes is inactivated, and you will talk about that later. Yes. So I will not talk about that, but that's an interesting fate. Instead, I will introduce you to an hypothesis, which I'm not quite sure it's true, but it's very intriguing. The idea is that there is a gender war here, and there are different interests from each partner, and the father would like a big Rambo boy that would fight well, protect family, while then the mother's evolutionary imperative would be her own survival, and to stay alive in order to take care of her litter and the next litter. And that would be a difference in interest. This is an interesting hypothesis, but it's not necessarily true, and I actually believe that this is more some kind of wrong thinking. But I would like to mention it anyway. It's called the parental conflict hypothesis. So coming back to the definition of sex, we have a genetic definition that is control, that's presence of the gene, but now we have introduced epigenetic, that is the use of the gene. And this is added another dimension to sex. And I like to mention, in fact, no one has mentioned this before at this conference, we have a 3rd gender, and only last year in Germany it was legally accepted as a 3rd gender, and in Sweden we have 100,000 births every year, and 15 to 20 of these children you cannot say which sex they have. So we need to then decide whether we want to define sex by the passport or by how the person conceive or perceive his or her life. So I think I missed something here, but I'm not quite sure what. All these are called primary sex-determining factors, and this leads to an irreversible differentiation of external genitalia and maturation of secondary sex characteristics like body, size, voice, etc. It's also quite clear that sex also specific sex development can occur also on non-reproductive, non-genital genitalial tissues and organs. We are not only talking about genitalia when we're talking about sex-specific differentiation. And so the question is, can sex influence cell fate? And the short answer is, can I go back? It should be yes. It should be appearing a yes. Can I go back? Previous? Oh, yes. At the bottom we have. Short answer is yes. Sex. Pardon? Yes, yes. Sex can influence cell fate, and hormones are the ones that actually do this. It means that cells from males and females can behave differently in physiological conditions, and there's no question about that. Can sex influence therapeutically relevant stem cell traits? And in your website that you showed yesterday, you showed some mouse work where female stem cells have different muscle generation, regeneration efficiency, and mesenchymal stem cells, that is from the bone marrow, also have a gender difference in mice. Now we can add from this lecture a human that we have the first report from bone marrow derived mesenchymal stromal cells in human, stem cell renewal and aging are difference. You can find differences, and these are the references that I found, and you can get them if you would like to know. But as a general rule, cells from males and females are the same. They differentiate, they divide, they differentiate, and they die. But then there is, on top of that, these differences. To sum up, the literature is sparse, definitely sparse. But together with the introduction I gave you of what's reasonable, it's very reasonable to suggest that cells from males and females can behave differently in physiological conditions. Sex can influence cell fate. Sex can influence therapeutically relevant stem cells traits, which is of importance when you think about regenerative medicine and to optimize conditions. And you can also have sex-specific development outside the sex organs, which is sometimes forgotten in the discussion. So in my first sum-up slide, I said, who is surprised? But I took that away and made this more formal summing up. So with that, I would like to end, but I also want to thank the Center for Gender Medicine, which I already did, actually, but I did promise you. So thank you very much for listening, and I'm happy to take questions.